Welcome to this instructional video to finish Chapter 12. Well, we've talked about previously adenylyl cyclases, cyclic AMP, and protein kinase A. Well, guess what? There's guanylyl cyclases, cyclic GMP, and protein kinase G. Let's talk about two different types of guanylyl cyclases, and the first is going to be membrane spanning. The second is going to be soluble nitric oxide activated guanylyl cyclases, and we're going to just forget that one. The first guanylyl cyclase, it's membrane spanning, and it is activated by peptide hormone called ANF. And let me write this out for you. If I can get this spelled right. So the receptor, the ANF receptor binds ANF, and then this is involved in vasodilation. Another type of guanylyl cyclase is it's not membrane spanning, it's not membrane associated, it's soluble in the cytosol, and it is activated by nitric oxide. And so those are the main two we want to talk about. In each case, a guanylyl cyclase is activated. And if you activate a guanylyl cyclase, it converts GTP to cyclic GMP. So let's talk a little bit about what cyclic GMP does. And then as a follow-up, we're going to talk more about protein kinase G. So just to recap on this page, there's membrane spanning guanylyl cyclases. The ANF receptor is one. The other are just soluble nitric oxide activated guanylyl cyclases. All right, well, let's talk a little bit more about protein kinase G and cyclic GMP. And there's some analogies here from protein kinase A and cyclic AMP. Protein kinase G is a single polypeptide. Now that's different than protein kinase A, recall. But protein kinase G is going to be regulated by cyclic GMP, much like protein kinase A is regulated by cyclic AMP. So this protein kinase G, even though it's a single polypeptide, no domains, there is a regulatory domain that blocks the active site of protein kinase G when cyclic GMP binds to the regulatory subunit you get a conformational change, and basically the active site becomes unblocked from the regulatory domain. It's open for business, if you will. Protein kinase G is a serine threonine protein kinase. And all I want to say about this today is that it has many targets. Now, we've talked a lot about the targets of protein kinase A, and when I say targets, that's the protein that protein kinase A binds to a protein and in an ATP-dependent manner phosphorylates a serine or threonine. Well, protein kinase G has a whole different set of targets that it can phosphorylate in an ATP-dependent manner and modulate their activity. All right, so we know cyclic GMP is made from guanylyl cyclases, and once cyclic GMP is made, it can bind to protein kinase G and activate protein kinase G. Now, so again, there's an analogy here, because cyclic GMP, much like cyclic AMP, is a second messenger. Let me just give you some broad functions of cyclic GMP in different tissues. So in the kidney and intestine, it's involved in changes in ion transport and water retention. Again, I'm just giving you some broad pieces of information here. Number 2, in cardiac muscle, it's involved in what we call, I guess, signal relaxation. Toning down the beating of the heart. And 3, the brain, it's involved in development and brain function. Development of the brain and in brain function. The one that I want to talk about the most here is on this next page is cardiac muscle, in terms of how is it involved in what we're calling signal relaxation. So let's talk about nitric oxide activated guanylyl cyclases. And to talk about that, we have to talk about nitric oxide. It's a very small molecule, an important molecule, and it's involved in signaling. And so how do we make nitric oxide? Well, arginine can be converted to citrulline, and nitric oxide is a product. I'm sure you're dying to know what the Lewis structure of nitric oxide looks like. Please don't confuse nitrous oxide with nitric oxide, because this is nitric oxide, NO, and the enzyme that does that is nitric oxide synthase. And so what we know is that nitric oxide is involved in a condition called angina pectoris. And angina pectoris is the pain associated, or actually pain caused, by contraction of heart muscle deprived of oxygen. And so how does this work? Well, we have a natural source of nitric oxide. There's also therapeutic agents that release nitric oxide. One of them is nitroglycerin. My great-grandmother had congestive heart failure, and she took nitroglycerin pills, and it breaks down to nitric oxide when you ingest it. The interesting thing is, remember, nitroglycerin is really dynamite. So there we go. So let's talk about this. What about the NO nitric oxide induced relaxation of cardiac muscle? So what we know about nitric oxide, let's talk about two things. Number one is that it is sufficiently nonpolar, and it can cross membranes quite easily. It's small, nonpolar. Hey, it can do simple diffusion. The second thing is it can activate soluble guanylyl cyclases. That's what we just talked about, right? So nitric oxide can activate enzymes, guanylyl cyclases, that are soluble in water. They're not membrane associated. And basically, if you activate soluble guanylyl cyclases, then what happens? Well, you're going to increase the concentration of cyclic GMP. If you do that, I guess we're on a detail train, buckle up, you're going to activate protein kinase G. If you activate protein kinase G, then you activate calcium ion pumps in heart tissue. These are responsible for the removal of calcium ions from the cytosol of heart tissue or heart cells, and then you basically decrease contraction of these muscles. So, how does nitric oxide induce relaxation of cardiac muscle? This is how we naturally make nitric oxide from arginine, but there are pharmaceutical agents like nitroglycerin that you can take that will break down and release nitric oxide to help, I guess, quiet heart activity. And so again, in this case, a nitric oxide is a small molecule. It's nonpolar. It can do simple diffusion. It can activate soluble guanylyl cyclases. We make more cyclic GMP that activates protein kinase G. Protein kinase G, one of its targets are calcium pumps. If you activate calcium pumps, which would be integral membrane proteins in heart muscle, then you remove calcium ions from the cytosol, and if you start removing calcium ions, you're going to decrease muscle contraction. All right. So I didn't say a lot about the, if we go back up here, I didn't say a lot about the Amf receptor, and I'm not going to at this point, but that's just one example of a membrane-spanning guanylyl cyclase. The one that we just went into detail about was soluble nitric oxide activated guanylyl cyclases. Again, both of these make cyclic GMP from GTP. Cyclic AMP binds to the regulatory domain of protein kinase G. It opens up the active site, and then protein kinase G, in an ATP-dependent manner, can phosphorylate a variety of targets. And the one target that we just talked about in, I guess, on page 31 was activating calcium pumps, which leads to the removal of calcium from the cytosol and decreased contraction. Well, it turns out that cyclic GMP is also involved in something you may hear a lot about on TV, Viagra and Cialis, which are drugs for erectile dysfunction. And so what we know here is cyclic GMP. Now recall that cyclic AMP can be broken down by a whole family of phosphodiesterases. Well, there's also an enzyme, a group of enzymes called cyclic GMP phosphodiesterases, and there's various isoforms of this. Now what that does is that converts cyclic GMP to just 5' GMP. And the enzymes are Cyclic Gmp, PDE, phosphodiesterases. Again, analogous to cyclic AMP phosphodiesterases. Well, now, here's the good information to know about this page. The isoform of cyclic GMP phosphodiesterase in the penis is inhibited by, and I'm not going to give you the, I guess, the technical name. I'm going to give you the common name, by Viagra and Cialis. I'm going to take one quick break. I'll be right back. All right, we're back. So, the isoform of cyclic GMP phosphodiesterase in the penis is inhibited by Viagra and Cialis. What is the consequence of this? Then you are going to increase the concentration of cyclic GMP. And because you are blocking an enzyme used to degrade cyclic GMP, if you block the cyclic GMP degrading enzyme, you are necessarily going to have a higher concentration of cyclic GMP. And the bottom line on this is that a higher concentration of cyclic GMP leads to an increased blood flow into the penis. And of course, this is going to help erectile dysfunction. All right, so if you've ever wondered how these drugs work, then now you know. They block cyclic GMP phosphodiesterase. Well, let's just tie up some loose ends. There's another important type of receptor system, cannabinoid receptors. And I'm not going to say a whole lot about this. We have cannabinoid receptors, and they are, wait for it, G protein coupled receptors. And they're pretty famous because this is what THC from marijuana binds to. And then there's obviously physiological effects that are a consequence of that. But we have endogenous, there's a great word, endogenous endocannabinoids. That's a good one. Endogenous endocannabinoids. And there's one that I want to call your attention to, and it's called anandamide. And I have I'll give this to you in class, supplementary notes, chapter 12, number 3 is going to be about anandamide, the kind of the endogenous feel-good molecule. And so what the supplementary notes will talk to you about is that there are certain enzymes called fatty acid amide hydrolases that degrade anandamide. And depending upon the genetic lottery in which you either won or didn't win, you may have more or less anandamide around, and that can believed to be kind of affecting your overall general mood. And then, of course, if you smoke marijuana or take marijuana in some form that provides THC, THC can bind to that same receptor and provide similar responses. Really, all I'm going to want you to know about page 33 is what's in supplementary notes, chapter 12, number 3, and about anandamide. All right, the last thing in this chapter is what we call receptor crosstalk. So let me write out a quote that's from your textbook that I think is pretty good. The textbook says the regulatory circuitry that governs metabolism, if I can write this better, is richly interwoven and multi-layered. And I think that's very true. So we've talked about, for example, the insulin receptor and the beta-adrenergic receptor. So I'll tell you what, let's not worry about this part right here. If you look at this right here, we talk about the action of the beta-adrenergic receptor is in opposition to the insulin receptor. And so how do they work together or talk to one another? Well, they can directly influence one another. That's why we say that the regulatory circuitry is richly interwoven and multi-layered because of this interaction. Let me go back and rewrite circuitry. I don't think I like that. Hang on a second. That's much better. So here we go. Here's an example of the insulin receptor, or insulin action, if you will, through the insulin receptor can actually oppose the metabolic effects of epinephrine. Because what this is showing you is that insulin binds its receptor, IRS1, it doesn't show it here, becomes phosphorylated through a series of events that we talked about in class. Protein kinase B is activated. One of the targets of protein kinase B is the cytosolic tail of the beta-adrenergic receptor, and then PKB can phosphorylate some serines. The actual insulin receptor tyrosine kinase can phosphorylate some tyrosines, so we get a mixing of phosphorylation events here. We get both serines and tyrosines phosphorylated, and then basically when this phosphorylation occurs, it promotes the internalization of the beta-adrenergic receptor. And so again, this is just one kind of small example of receptor crosstalk. Insulin action can basically shut down work or activity at the beta-adrenergic receptor because of this phosphorylation initiated receptor internalization. That's pretty much what this is showing us. So don't think these things work independently of one another, because they really don't. There is some crosstalk between them. All right, that's it for chapter 12. Thank you.